Resistance and the immune system. Acquired immunity. Acquired immunity defends against a tremendous variety of potential pathogens. This defense is based on 4 important attributes. Number 1, specificity. Number 2, tolerance of self. Number 3, minimal self-damage. And number 4, immunological memory. Innate immunity has the ability to recognize a diversity of foreign substances. They are capable of mobilizing the specific acquired immune system and provoking an immune response by B cells and T cells. The acquired immunity usually involves protein receptors on B and T cells that recognize discrete regions of the antigen called antigenic determinants or epitopes. Each different antigen is capable of stimulating an immune response. Under normal circumstances, one's own cells and molecules with their own molecular determinants do not stimulate an acquired immune response. This tolerance occurs because the epitopes are interpreted as self. A population of T cells, called regulatory T cells, prevents autoreactive T cells from attacking self. In fact, those T cells recognizing self are normally destroyed through apoptosis. The immune system thereby develops a tolerance of self and remains extremely sensitive to non-self antigens. Antigens present on pathogens, tumor cells, and cells of other individuals of the same species. Should self-tolerance break down, an autoimmune disorder such as lupus may occur. In an acquired immune response, it is important that the response be strong enough to eliminate the pathogen, yet controlled so as not to cause extensive damage to tissues and organs of the body. In fact, many of the symptoms of an infectious disease are due to the innate and acquired immune responses, and not directly from the pathogen. Sometimes the immune system extremely overreacts in a potentially fatal condition called anaphylactic shock. Most of us during our lives have become immune to certain diseases, such as chickenpox and measles, from which we have recovered or to which we have been immunized. This precise long-term ability to remember past pathogen exposures is called immunological memory. Immunological memory is a hallmark of acquired immunity. Thanks to immunological memory, a second or ensuing exposure to the same pathogen produces such a rapid and vigorous immune response that the person does not even know that they were exposed. The cornerstone of acquired immunity are the lymphocytes. The 2 types of lymphocytes are B lymphocytes and T lymphocytes. The B lymphocytes, also known as B cells, are involved in the process by which antibodies are produced against specific epitopes. The T lymphocytes, also known as T cells, provide resistance through direct cell-to-cell contact with infected or otherwise abnormal cells, and then terminate these cells by lysis. Immune recognition only activates the appropriate B and T cell clones that recognize non-self epitopes. B cells have antibodies on their surface that perform receptor functions by binding to an antigen or epitope. T cells have an antibody-like receptor that binds antigenic fragments. In clonal selection, only those B cells that have a surface receptor that matches the shape of the antigenic determinant will be stimulated to divide. This division produces clones of identical B cells. Lymphoid organs contain many naive B cells whose surface receptors, or antibody molecules, have unique specificity. Binding of an antigen by an appropriate B cell receptor and chemical stimulation from a T cell trigger B cell activation. B cells undergo cellular division to form a clone of activated B cells. The activated B cells then secrete cytokines. These cytokines cause B cells to differentiate into plasma or effector cells and memory B cells. Plasma cells secrete antibodies into the circulation. These antibodies recognize the same epitope originally bound to the B cell receptors. Lymphocytes arise in the bone marrow and mature either in the bone marrow as B cells or in the thymus as T cells. Humoral immunity is a response mediated by antigen-specific B lymphocytes. Antibodies are a class of proteins called immunoglobulins. Immunoglobulins react with epitopes on toxin molecules in the bloodstream, on antigen surfaces or microbial structures, and with epitopes on viruses in the extracellular fluid. Antibodies recognize specific shapes associated with an antigen's epitopes. An immunoglobulin monomer consists of 2 light chains and 2 heavy polypeptide chains. The variable domains in a light and heavy chain form a pocket called the antigen binding site. Here is a molecular model of the antibody immunoglobulin G, or IgG. There are 5 classes of immunoglobulins. Using the abbreviation Ig for immunoglobulin, the 5 classes are designated IgG, IgM, IgA, IgE, and IgD. There are both primary and secondary antibody responses. After the initial antigenic stimulation, IgM is the first antibody to appear in circulation. Later, IgM is supplemented by IgG. On a subsequent exposure to the same antigen, the production of IgG becomes more rapid and the concentration in the serum reaches a higher level than in the primary response. The first time the body encounters a pathogen or antigen, a primary antibody response occurs. B cells are activated. The effector B cells and plasma B cells start producing and secreting antibodies. A second or subsequent infection by the same pathogen or antigen produces a more powerful and longer-lasting secondary antibody response. Due to the presence of memory cells, a rapid response to antigens leads to the production of IgG, which is the principal antibody. The secondary antibody response occurs faster and is more vigorous than the primary response. The secondary antibody response also provides a much longer period of resistance against disease. Embryonic cells contain about 300 genetic segments which are shuffled like transposons and combined in each B cell as it matures. This process is known as somatic recombination. Current evidence indicates that more than 600 different antibody gene segments exist per cell. In addition, diversity is generated through imprecise recombination and somatic mutation. Therefore, the total antibody diversity produced by the B cell is in the range of 10 to the 14th power immunoglobulin possibilities for the variable region of the antibody. In order for specific resistance to develop, antibodies must interact with antigens so that the antigen is changed in some way. Thus, the formation of antigen-antibody complexes may result in death to the microorganism possessing the antigen, inactivation of the antigen, or increased susceptibility of the antigen to other body defenses. Antibodies react with molecules at the viral surface and prevent viral attachment to cells. Antibodies combine specifically with toxins or microorganisms, thereby neutralizing them and preventing attachment to cells. Antibodies called opsonins coat bacterial cells, preventing bacterial attachment to cells. Antibodies combine with antigens on the surface and bind the cells together or restrict their movement. Antibodies combine with dissolved agents to form lattice-like arrangements that precipitate out of solution. The Fc portion on antibodies encourages phagocytosis by forming a bridge between antigen and receptor sites on the phagocyte. The body's defense against microorganisms infecting cells and other non-self cells is centered in the cell-mediated immune response. This is done by the formation of a membrane attack complex, or MAC, through the activation of a complement cascade. These MACs are formed in the cell membrane of a pathogen. For example, IgG or IgM attaches to the antigens on the pathogens located on the pathogen's cell membrane. Complement proteins then link antibody molecules together. Activated complement proteins attach to the pathogen's membrane in a step-by-step sequence, forming a membrane attack complex, or MAC. The attack complex results in pore formation or hole formation in the membrane, causing cell lysis. B cells and T cells are both lymphocytes, but they behave a little bit differently from one another. B cell receptors directly recognize epitopes on an antigen. whereas T cells contain surface receptors that are capable of recognizing a multitude of antigenic fragments due to the process of somatic recombination. Some T cells are effector T cells. There are two major subpopulations of effector T cells. The T cell's receptors determine how the cells function in cellular immunity. There are cytotoxic T cells and helper T cells. Cytotoxic T cells gain the T cell receptor and the CD8 co-receptors that recognize antigen peptides located on virus-infected cells and tumor cells. Helper T cells gain T cell receptors and CD4 co-receptors. One class of helper T cells help to eliminate pathogens from infected antigen-presenting cells, and another class of helper T cells help to activate B cells and antibody production. T cells that recognize self are destroyed through apoptosis. The actual acquired immune response originates with the innate immune response and the entry of antigens into the body. At the site of entry, or within the lymphatic system, the antigens are phagocytized by the antigen-presenting cells, which are dendritic cells or macrophages. As these cells migrate to the lymph nodes or other secondary lymphoid tissues, these antigens are broken down and peptide fragments are displayed on the surface as MHC2 peptide complexes. When the antigen-presenting cell is bound to the T cell, it secretes specific cytokines such as interleukin-1, which binds with naive T cells and stimulates T cell activation. Interleukin-1 causes the naive T cell to secrete other cytokines, including interleukin-2, which stimulates cell division of that T cell and others activated by the antigen-presenting cells. The result is the production of a clone of antigen-specific activated T cells. Some of these T cells will mature into memory T cells. Memory T cells will await for any future encounter with the same epitope and launch an immune response. Antigen-presenting cells, such as dendritic cells or macrophages, activate naive T cells. The activated immature T cells stimulate humoral immunity and stimulate the destruction of intracellular pathogens. This process also helps to activate cytotoxic T cells. These cells secrete proteins like perforins and granzymes that kill virus-infected cells or cancer cells using apoptosis. Antigen-presenting cells, or APCs, locate the appropriate naive CD4 and CD8 T cells and activates them. Interleukin-1 from the antigen-presenting cell augments the activation of CD4 cells, while interleukin-2 further stimulates CD8 cells. The activated CD4 and CD8 T cells divide to give rise to a clone, all recognizing the same antigen peptide. The cells secrete interleukin-2, which further increases cellular division. Some memory T cells will form from the division of the activated T cells. Memory T cells will be distributed to the tissues for long-term immunity. Cytokines stimulate the maturity of helper T cell populations. Lysosomes fuse with the phagosomes, destroying these pathogens. The cytotoxic T cells divide and form clones. These cells leave the lymphoid tissue and travel to the site of infection. At the site of infection, these cytotoxic T cells bind to antigen peptides that are enclosed within the MHC1 proteins. The cytotoxic cells release perforin and granzymes, which enter the infected cells and destroy them. Secretion of specific antibodies from plasma B cells depends on B cell presentation of the MHC2 peptides, as well as the binding of Th2 activated cells to the MHC2 peptide complex. The antigen receptor complex has been taken into the selected B lymphocyte, and the antigen peptide is now displayed on the B cell's surface within the class II MHC protein. A clone of activated Th2 cells results from cell-mediated immunity. The activated Th2 cells bind with the selected B cells. The binding takes place at the MHC2 peptide complex. This binding spurs the B cells to enlarge and divide into a clone of plasma B cells and memory B cells. The plasma cells secrete antibodies that enter the body's circulation and find their way to antigen-bearing microorganisms. Humoral immunity produces antibodies that respond and bind to antigens. Cell-mediated immunity stimulates helper T cells that activate B cells and cytotoxic T cells. Cytotoxic T cells bind to and kill infected cells or otherwise abnormal cells. Memory B and T cells are important in a secondary exposure to the same antigen. Thank you for watching.